All right, we're gonna get started. Okay, so even though Greg kind of pointed out some of the pitfalls of the parametric modeling in his talk, we're gonna go back to the parametric modeling now and revisit how it's done in a little bit more detail. I'll show you some examples, and that should take about 20 minutes or so. And then hopefully around 11 we'll transfer over to the lab. I'll have a quick lab session before lunch where you guys will actually train a model so that it can train over lunch, because these things take time. So one of the reasons we might want to go back to the parametric model is the diffeomorphic models are actually quite computationally intensive. So it takes a long time for both training and for synthesis. So if you don't have a lot of time to sit around and wait for these things to happen, the parametric models are often a much more attractive approach. All right, so just to review for either approach, what you need in Cell Organizer right now are fluorescent microscopy images. Currently, we don't support any other type of imaging when it comes to raw images, at least. And those images need to have at least a nuclear marker, and then a cell marker, a protein marker, and potentially a cropped region of interest. So I'll show you in a second, but a cropped region, if you have like a field of cells where you have a lot of cells, is really useful for saying which cell you want to look at so your algorithms don't get confused. And then you need to tell Cell Organizer what type of protein you're modeling and what resolution you're modeling it at, and a couple other things that we'll look at in the lab later on. The protein type is important mainly for housekeeping reasons, so that in the future, if you or someone else that is trying to use your model is trying to look for, I want a model of endosomes, or I want a model of lysosomes, they can do that. Or in our case, say I give Cell Organizer an SBML file and I say, hey, please create the required geometries for me. If you don't tell it what type of protein, what class your protein is in, what organelle, it won't be able to find that model for you. So the model will be kind of useless. And the resolution is really important because although we haven't really discussed it too much, Cell Organizer actually supports training these models at multiple resolutions and then synthesizing them all together in a single image. So if you have one experiment at 100x and another experiment at 40x on your microscopes, you can actually train models at 100x, train models at 40x, and bring those two models together. And so then in the output, you'll get some sort of model, and that'll allow you to make these synthetic images that we've been seeing and started to play with yesterday. Okay, so let's look at what this sort of looks like. Here's an example microscopy image, and you can see in blue we have our cell. This is a cytoplasmic marker, although you can also use a plasma membrane marker to get the cell. In red we have the nucleus, and in green some fluorescent protein. And you can see it in this particular image, it's relatively clean. There's pretty much only one cell, but there are these pieces here, so just a simple box for a cropped region is fine. If you had a lot of crowding, you might want to think a little bit harder about a cropped region. If you don't provide Cell Organizer with a cropped region, it's okay. It's just going to find the largest object that it can in the cell channel and call that the cell. So this is something to be aware of. It's not going to pick your favorite cell. It's going to pick the biggest cell. And if two cells are touching and overlapping and it segments that into one object, it's going to think that's a cell. So the cropped regions in the cases where you have cells that are really close together, it's a really important thing to go through and do. So when I say required, I should clarify, if all you care about is building a nuclear model, the only thing that's required is a nucleus. If you care about building a cell model, currently in Cell Organizer, you need a nucleus and a cell marker because of the way the cell is being parameterized. We're hopefully going to separate some of these compartments so that they can be learned in a conditional manner and in an independent manner in the future. But for right now, the ordering here is intentional. That is, if you want a nucleus, you can do just a nucleus. If you want a cell, you have to do the first two. And if you want to figure out where model a protein inside a cell, you need both the nucleus and the cell. Okay? So a lot of you are maybe not in the field of microscopy, or even if you are, it's a good idea to take a look at some of these resources. These slides are going to be published online for you guys to look at later, so don't worry about scribbling this down. But these are just some resources that you can go to that have these fluorescent image databases for you to start looking at and seeing if you can find images that match your problem that you're interested in. Of course, the top one there, experimental work, is the best way to get something that you know is going to be the system that you want it to be and the conditions that you want it to be under. Anytime you're using someone else's data, you kind of have to be really careful that you are absolutely aware of how they set up their experiment and everything like that. Okay, so Cell Organizer has a built-in segmentation method. So basically, we start out with, if it's the first slice, we just start out with the cropped region, or if you don't have a cropped region, the entire image. If it's a subsequent slice of a three-dimensional stack, so a Z-slice, we just start out with the mask from the previous segmentation, the previous slice's segmentation. And then we perform active contour. So this is a little movie of what happens when you active contour. So basically, you're starting with some boundary, and you're shrinking it and looking for the gradient in the intensity of the image. And that runs for some number of iterations until either it converges, that is, the boundary isn't moving very much, or it reaches the maximum number of iterations that we allow in Cell Organizer. And then it goes to the next slice, and it keeps doing that. There's also an option in Cell Organizer, which I strongly encourage you to use if you have your own images, and that is that you can pre-process your images outside of Cell Organizer. You wouldn't want to do this for the protein images because of the way we're fitting a Gaussian mixture model, but for the cell and nucleus, where we're fitting shape models to them, having your own segmentation algorithm is really useful because segmentation is an ill-defined problem and changes a lot depending on what you are trying to look at or what type of cell you're segmenting. So we make no guarantees about how great our segmentation method is. It's just the one that works for us, and you're welcome to use it or give us pre-segmented images, and Cell Organizer will work on those too. Okay, so when I said nuclear images were required, that was a bit of a lie. Greg has done some really cool work to try to get around that sort of pitfall of Cell Organizer, because a lot of times you don't have a nuclear image. So what you can actually do in Cell Organizer is just pass it an image. Say it's a cytoplasmic marker, maybe it's just a protein that takes up a large amount of the cell. You can give Cell Organizer that image. So this is like a intensity. So higher color, or warmer colors are higher intensity here, and lower colors are less intense, less protein intensity. And you can give it that, and it will threshold that image, find a convex hull, and then it's going to try and find the nuclear signal where there is no signal, basically. So if you have a cytoplasmic protein that's just ubiquitous in the cytoplasm, or even if it's in some organelle, if it's reasonably reasonably dense throughout the cell, this method will work, because you'll end up with a large region inside your cell that there's actually no signal. And so what this method does is it tries to find that big hole and fit a nuclear shape to it, and then from there you can continue on with your modeling process. Okay, so how do we build the nuclear model? So we take the nucleus, or if you don't have a nucleus, we're going to find the hole, and we get some segmentation of it, and then we orient that along its major axis. And then in the 2D case, we build a medial axis model. So going down the center of the cell, we're measuring the distance from the medial axis. Or in 3 dimensions, like Dr. Murphy talked about, we build a 3D spline model, and he described how we sort of unwrap that in a cylindrical coordinate system and then smooth it with a spline. And we learn the parameters of those splines for a bunch of nuclei, and then we can synthesize new nuclei from that model. Okay, once we have our nuclear model, we can build a cell model. And so the way we do that is, again, we segment our cell using active contouring. This is what a cell segmentation looks like here on the bottom for the original cell that I'm showing here, here on the top. And if you have an adherent cell line, you might tell a cell organizer, I have an adherent cell line. There's an option for doing that. And cell organizer will cut off the bottom of the cell so that your cell is actually flat. And it's not really clear on this particular cell, but in a lot of cases, if you are imaging a cell, you'll image a little bit below and a little bit above your cell in order to make sure that you get the whole cell in the image. However, when you're automatically pre-processing this, if you're outside of the focal plane of the cell, but sort of still near it, you'll get some fluorescence from the cell that's not actually, it's out of focus fluorescence, out of plane fluorescence. And so if you know that it's adherent, you know that basically the largest slice of the cell is going to be on the bottom. So in some cases, some of these bottom slices can tail off much the same way that the top slices do into almost nothing as you go down into the cover slip, and you don't want that to be considered part of your cell. It's an inaccurate representation. So you can tell Cell Organizer that, and it will do that for you. Or you can provide, again, your own segmented cells and avoid all of these issues entirely. Okay, so once we have our cell segmentation, we again orient it, and then we start building our cell model. And I think Bob described a little bit about how we do this. It's a ratio-based model. So for each angle here that's marked with theta, we're going to look out from the center of the nucleus to the edge of the cell, and we're going to get some distance, and then we're going to look from the center of the nucleus to the edge of the nucleus along that same ray and get another distance, and then model the size of the cell at each angle theta with a ratio of those two distances. Yeah? Okay, so the way that's done, basically in visual form, it's a little bit hard to see here, but if you look, you can see we're actually going around the cell at each point and picking out where we're hitting, and each of those is being measured as a distance. So this is the reason I'm showing you this is not because I think it's super informative for you to see that for this particular case, but it's an output that you can have Cell Organizer make for your cells, and it's useful if you want to look to see that the segmentation and the representation of the cell is good. So in the case where you have a neuron like Greg showed, the hit points would not capture the neurites, and using this visualization in Cell Organizer as you're going around, you would be able to see that you had suddenly skipped a section of the cell that you were actually interested in. And the way that you could correct that is to either use the non-parametric method or maybe sample a smaller theta as you go around. The default is 1 degree, so you get 360. Okay? So once you've built that ratio model of the cell, we have what we call in the Cell Organizer development community the cell framework model. That just means that we have some cell that we can now fill with proteins. So we're ready to try and fill out the rest of this cascade here. We're going to ignore, for this talk, the microtubule model, since that part is not currently supported in the simulation modeling. But for the vesicular organelle model, we start again with a protein labeled image. This is just some central slice from a sample image that I chose. Again, this is the case where you don't want to give your own pre-processed data, probably. If you give a black and white image of your protein, we're not going to be able to fit accurate objects to it. So you'll start out with this raw image here, and using Cell Organizer pre-processing, we'll get only what we think are relevant objects. So you can see here on the left, we have a lot of signal that's not actually really a vesicle, it's just autofluorescence within the cell, or maybe that protein isn't always necessarily localized to your organelle, and we want to get rid of that. So now we have our preprocessed image, and then we mask out our objects. And again, remember that this is actually happening in 3D, but once we mask out our objects, we take each contiguous object within the cell and we pull it out and we fit our Gaussian mixture model. So you saw this slide when Bob was talking. This is an example object. This is actually one contiguous object, and you can tell that it's actually not one vesicle just by looking at it. So we apply a filtering to it, and then we can fit it with Gaussian. So on the left is the one that Bob showed. That's actually using spherical Gaussians, and on the right is using the full covariance. So in other words, we allow now elongated Gaussians to be fit, so that not all your vesicles have to be spherical. All right. So what that gives you is some number of these Gaussians that you've now fit, and their relative size and intensity. And like Bob talked about, we can then place these within the cell by modeling their relative distance in between the edge of the nucleus and the edge of the cell, and building a probability density map. Okay, so now we have some wholesale model. We can synthesize either nucleus and cell, or all three, nucleus, cell, and protein. And you've seen some of these things before. Like on the far right, you've seen what we've been using in MCell. Cell Organizer also has several other output modalities in addition to the SPML spatial. We can output just simple TIFF images, or what we call sampled images. So TIFF image, it's just going to be a three-dimensional image where each slice is represented as a separate image. And in this case, organelles are represented in blue or green, depending on the type of organelle. So again, we've learned two models now. For cell shape and nuclear shape, we learned a single model, but two models of protein localization from two separate data sets, potentially learned at different resolutions, and we can then synthesize within a single framework what it will look like if you had both of these proteins inside of one cell. So that's TIFF images, and then sampled images might not seem super useful for running simulations, but this is meant to mimic what you might see if you were actually on a microscope looking at these cells, and this is useful for other applications. I won't get into too much detail, but basically the idea is with some of these segmentation tasks that we try to do, there's currently no ground truth for what a good segmentation is versus a bad segmentation. You just have to look at it. So the idea behind recapitulating something that looks like a microscopy image with these generative models is that we can then build methods to try to learn what the optimal parameters for a given segmentation algorithm are, and then map those learned segmentation parameters onto some feature space of the image and use that to segment the image in an optimal way, or at least an intelligent way. Okay, so that's really all I wanted to discuss before we actually get into the hands-on section of the lab. Does anyone have any questions about how training works, what we're going to be doing in the lab, some of these modeling techniques? Everyone gets it? All right. Well, in that case, without further ado, let's run over to the lab and get started training some models. Thanks.